Hello everyone, welcome to our new lecture. We are learning chapter number 10, that is cell and cell organelles. In our previous lecture, we have seen about the diffusion and osmosis which is carried out between the cells. Now, let us study about the cytoplasm. So what is cytoplasm? It is the fluid between the plasma membrane. Okay, so between the plasma membrane of the cell and the nucleus, you will find this particular part of a cell, that is cytoplasm. And what it is a fluid. Okay, क्या है ये? एक fluid है जो plasma membrane और nucleus के बीच में होता है. It is a moving sticky substance. Okay, it moves and it is a sticky substance. Many cell organelles are suspended in cytoplasm. We have to keep in mind that in the cytoplasm you will find all the cell organelles, okay, of a cell. Cytoplasm is the medium for cellular chemical reactions. So whatever chemical reactions that are taking place in the cell are carried out in the cytoplasm. Okay, so all the chemical reactions which is carried within the cell is done or that takes place in the cytoplasm. Okay, now the part of cytoplasm other than organelles is the cytosol. Okay, keeping the organelles of the cell apart, the rest of the part in a cytoplasm is called as cytosol. Okay, now cytosol stores vital substances like amino acids, glucose, okay, vitamins, etc., etc. So all these important vital substances which can be used to create energy like amino acid, glucose and vitamins, all these important substances, important compounds are stored within the cytosol. Okay, now in animal cells, cytoplasm is more granular and dense, while in plant cell it is thin and mostly pushed to the periphery due to a large central vacuole. Okay, now the difference between the animal and plant cells cytoplasm is given over here. Okay, that you will see in animal cells cytoplasm is more granular and dense. Okay, so the density of the cytosol in the cytoplasm of the animal is dense. Okay, whereas in the plant cell you will see that this cytosol is not that much dense in the cytoplasm of plant cell. Okay, and you will also see that mostly it is pushed to the periphery due to a large central vacuole. Okay, so this is about the cytoplasm. Now inside the cytoplasm there are a lot of organelles which are called as cell organelles. Okay, so the organs of the cell which is there within the cytoplasm, okay, they are called as cell organelles. Now, an organelle is a specialized subunit having specific function within the cell. What is an organelle? It is a specialized subunit having specific function within the cell. Okay, so एक ऐसा पार्ट किसी भी सेल का जो बहुत ही इंपॉर्टेंट पार्ट है, एक सब यूनिट है सेल का, और वो क्या करता है, एक स्पेसिफिक फंक्शन जो है वो परफॉर्म करता है सेल में, उसकी एक जिम्मेदारी होती है और वो जिम्मेदारी को पूरा करने का काम उसका होता है. ओके, दे आर ऑर्गन्स ऑफ द सेल. ओके, they are nothing but the organs of the cell. Now each organelle has its own lipoprotein membrane. Okay, so each of the cell organelle is going to have its own lipoprotein membrane. A very important sentence this is. Okay, and a very important word, lipoprotein membrane. So this is the membrane which is there on each and every organelle which is there, which is present in the cytoplasm. Now, except nucleus and chloroplast, all other organelles can be seen only with the electron microscope. Keep this in mind that we can observe the cell organelles with the help of electron microscope only. Okay, so keeping the nucleus and chloroplast apart, other cell organelles like endoplasmic reticulum, Golgi complex, okay, ribosomes, all these other cell organelles can be seen with the help of electron microscope only. Okay, now let us see one by one each and every cell organelle. So the first is nucleus. Okay, we can perform a simple activity over here. Take a drop of water on a clean glass slide. Using an ice cream spoon, gently scrape the inner side of your cheek. With a needle, transfer a little material from spoon to the water drop on the slide and spread it evenly. Put a drop of methylene blue stain on the smear. Put a cover slip and observe under microscope. Did you observe the cells with blue nucleus? Okay, now this is a very simple experiment where we can observe nucleus of a particular microorganism, okay, or a cell, and from where we are going to extract this cell, we are going to extract it from our own cheek. Okay, so हमें क्या करना है, एक स्लाइड के ऊपर क्लीन ड्रॉप ऑफ वाटर जो है वो डालना है, उसके बाद स्पून लेना है, आइसक्रीम का स्पून, उसे हमारे चीक के ऊपर बहुत ही जेंटली क्या करना है, स्क्रैप करना है, मतलब घसना है, और उस पे से विद द हेल्प ऑफ निडल जो भी उसके ऊपर सब्सटेंस होगा जो हमें दिखेगा या पता नहीं, लेकिन वो हमें क्या करना है, उस निडल की मदद से हमें वो वाटर ड्रॉप में डालना है। ओके, और फिर हम लोग उस ड्रॉप जो डाला है वाटर ड्रॉप, उस पर हम लोग मिथिलीन ब्लू का स्टेन डालेंगे। ओके, मिथिलीन ब्लू एक केमिकल है, वो केमिकल को हम लोग क्या करेंगे, मिक्स करेंगे वो ड्रॉप के साथ, और फिर हम उस पर्टिकुलर स्लाइड को ऑब्जर्व करेंगे अंडर अ माइक्रोस्कोप। और जब हम लोग उसे ऑब्जर्व करेंगे तो हमें क्या देखने मिलेगा, एक ब्लू कलर का न्यूक्लियस वाला सेल देखने मिलेगा। ओके, ऐसा सेल जिसका जो न्यूक्लियस है वो ब्लू कलर का है। नाउ द dark round spot seen under the microscope while observing the onion peeling stain with iodine is the nucleus of the cell. सेम हम लोग प्रोसीजर कर सकते हैं ऑनियन के रूट, जो ऑनियन का जो रूट होता है उसको अगर हम लोग बहुत ही जेंटली कट करते हैं, बहुत ही बारीक पीस में, बहुत ही थिन क्रिटिकल में, और उसको जब हम लोग ऑब्जर्व करेंगे अंडर अ compound microscope, okay, we have to definitely treat it with the other chemicals like HCl and methylene blue, and then we can observe it under a microscope. What we are going to do, we are going to see nucleus of that onion cell. Okay, root of the onion plant. Okay, now this is the figure of nucleus you can see here. Okay, what are the parts of nucleus? Chromatin, nucleolus, nuclear pore, and nuclear membrane. So these are the parts in a nucleus. Now when observed under electron microscope, we can see nucleus covered by a double layered membrane with small pores. These pores allow the passage of material in and out of the nucleus. Now nucleus has one round nucleolus and a network of chromatin fibers. Chromatin fibers are thin thread-like structures which condense to form short, thick chromosomes at the time of cell division. Functional segments on chromosomes are called genes. Okay, so nucleus क्या है, देखो जब हम लोग उसको इलेक्ट्रॉन माइक्रोस्कोप के अंदर ऑब्जर्व करेंगे तो हमें उसके कुछ पार्ट्स देखने मिलेंगे। ओके, सबसे पहला क्या है कि जो न्यूक्लियस होता है वो डबल लेयर्ड मेम्ब्रेन से कवर्ड होता है। ओके, दो लेयर का उसका मेम्ब्रेन होगा। ओके, दैट इज न्यूक्लियर मेम्ब्रेन, और उस मेम्ब्रेन के ऊपर आपको बहुत सारे स्मॉल पोर्स देखने मिलेंगे। ये पोर्स क्या करते हैं, जो भी इंपॉर्टेंट मटेरियल है वो न्यूक्लियस के अंदर क्या करते हैं, ट्रांसफर करने का काम करते हैं। ओके, उसके बाद न्यूक्लियस के पास क्या है, न्यूक्लियोलस प्रेजेंट होता है, और न्यूक्लियोलस क्या है, इट इज अ नेटवर्क ऑफ क्रोमैटिन फाइबर्स। ओके, सो क्रोमोसोम्स के जो फाइबर्स होते हैं, क्रोमैटिन के जो फाइबर्स होते हैं वो न्यूक्लियोलस के ऊपर आपको देखने मिलेंगे। ओके, क्रोमैटिन फाइबर्स आर थिन स्ट्रेट लाइक स्ट्रक्चर्स विच कंडेंस टू फॉर्म शॉर्ट थिक क्रोमोसोम्स एट द टाइम ऑफ सेल डिवीजन। जब सेल डिवाइड होता है, ओके, एक सेल से जब दो सेल्स क्रिएट होते हैं, उस टाइम पे ये जो क्रोमैटिन फाइबर्स है वो कन्वर्ट होते हैं क्रोमोसोम्स में। ओके, और क्रोमोसोम्स क्या है, क्रोमोसोम जो है वो बहुत ही इंपॉर्टेंट फंक्शनल सेगमेंट है न्यूक्लियस का, जिसे हम लोग जीन्स कहते हैं। क्या बोलते हैं, जीन्स, एंड इट इज वेरी इंपॉर्टेंट, वेरी मच इंपॉर्टेंट कंसिडरिंग द बायोलॉजी। दिस पर्टिकुलर पार्ट ऑफ द सेल, दैट इज जीन, इट इज वेरी 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 मच इंपॉर्टेंट। Now, what are the functions of nucleus? The first function, it controls all metabolic activities of the cell and also the cell division. So what is the first function of nucleus? It is used to control the metabolic activities which is performed within the cell and it is also responsible for the proper cell division of a cell. Okay, then next function is it is involved in the transmission of hereditary characters from parents to offspring. So whatever characteristics which are there in the parent, okay, those characteristics, those adaptations have been transferred to the offspring, to the child, by this particular organelle, that is nucleus. Okay, so these are the functions of nucleus. Now do you know that if the nucleus is lost or it is loosened from the cell, what will happen? The large quantity of hemoglobin can be accommodated in the RBC, and due to that a very large amount of oxygen can be transported to the cell. Okay, if that happens, definitely the amount of energy which will be produced by the cell will be more. But this is not a good idea, as nucleus is a very important organelle and it is responsible for one of the most important functions of a living organism. Okay, and these functions are actually responsible for the existence of that species. So nucleus cannot be loosened out from the cell in any case. Next is, due to the loss of nucleus of sieve tubes of the plant phloem, they become hollow, and thereby transportation of food becomes easy. Now if the nucleus is lost from the sieve tubes of a plant, okay, phloem, so phloem is a tissue which is responsible for the transfer of uh, food, okay, transfer of food within the plant. So if the nucleus is lost from these tissues, okay, from the cells of these tissues, they will become hollow, and a large amount or a very easily food can be transported within the plant. Okay, so these are some interesting facts about the nucleus. Now in this lecture we have seen what is cytoplasm, and we have also gone through the concept of nucleus. In the next lecture we will study about the different organelles, okay, which are there in a cell. So for this lecture your homework will be, you have to draw this diagram of nucleus in your notebook. Okay, now we will meet in the next lecture. Until then, goodbye.